Well, thank you, Vic, and thank you for hosting me to my first visit to Google, and I hope it's not my last. What I want to do in my talk is not really focus on science. And what I really want to talk about is how did I get to do what I did? And it wasn't like I woke up someday and said, I'm going to cure cancer. The reality is that there were lots of steps and lots of processes and lots of things had to fall into place. And as I look back, I can try and dissect some of those things. And you can apply success in any field to success in any other field. And so I hope what I'm talking about are maybe some life lessons, maybe some things that you probably already know, and I'm just telling you what you already know. So I'm just going to take you through some of those steps, step by step, of some of the things that I've learned about what had to come together for me to go from a laboratory to a clinical project and ultimately success in patients. So the first lesson is breakthroughs take time. As I said, you don't wake up some morning and some aha moment and you're all done. And there's oftentimes a very, very lengthy process. And the best way I can convince you that breakthroughs take time is to try to transport you back 100 years. Let's go back 100 years, early 1900s. Life expectancy in the United States is about 47 years. And the leading cause of death in the United States are infections. Pneumonia, tuberculosis, diarrheal diseases, infectious diseases are the leading causes of death in our country. But you go forward a century, and pneumonia, influenza, they make headlines now when a new infection comes along. When Ebola comes along, it makes headlines. HIV in the 80s made headlines because these were new infections and they were frightening. But by and large, we don't worry in our daily lives about infectious diseases. We worry about heart disease, cancer. You know, when you think about interactions with your doctor, it's you go to your doctor, you've got cancer, will I ever get cured? And those are the kinds of interactions that we have. So what happened over the course of the last century? What breakthroughs occurred to lead to the state of affairs we have with infectious disease, where we think of many of them eradicated, many of them treatable? And things like 1903, we actually started doing sewage treatment, chlorination of water, pasteurization of milk. In the 1940s, we put refrigerators in our houses. So these are the things we take for granted that make our food and water supply safe. 1940s, antibiotics. If you had said to somebody, I'm going to specifically target bacterial infections in 1900, they would have thought you were crazy or came from a different planet. So it wasn't until the 1940s that penicillin came along. And then the 1950s, vaccines. Polio vaccine was 1955. Smallpox was eradicated. And now in the news, it's people who aren't getting vaccinated. It's in the news. So we know vaccines work. But if you recast that, that's prevention. That's specific treatments, and that's modulating the immune system. So I could go through all of these breakthroughs, but again, this is a century of progress against infectious disease. So breakthroughs certainly take time. Now, the disease I worked on, called chronic myeloid leukemia, started with its first description in 1845. We understood the cause by about 1985, and by 2001, we had a specific therapy. So that's the timeline for the disease I worked on. The other thing that's important about this is what I call my translational research slide. You describe a clinical entity, you understand it, and then you can actually do something about it. So, clinical description of CML. 1845, two pathologists, Bennett and Virchow, described the autopsy findings of patients with leukemia. Now, let's go back to 1845. No microscopes. They did this based on autopsy findings, and then they submitted these articles for publication, and thereafter ensued a rancorous debate about who is really first, because these were published within 6 weeks of one another. So debates about priority, nothing new. But think about publishing in 1845. And some of you in this audience have probably grown up in a generation where there haven't been computers. In 1845, there weren't even typewriters. And just to give you an idea of what the current generation is growing up with, my daughter is 8 years old, my youngest daughter, and she just recently achieved a black belt in Taekwondo, and as a reward, she wanted me to buy her a typewriter so she could type out old-fashioned Google Docs. That was her impression of the world. She has not lived in a world without Google. But 1845, these two were typeset, submitted somehow to some central place for review, and ultimately they got published. And in a public lecture, Virchow said that Bennett had described his case 6 weeks before, so Bennett gets credit for first. So we'll fast forward about 150 years, and the disease I worked on, called chronic myeloid leukemia, is one of the 4 common types of leukemia, making up about 15 to 20% of all leukemias. There are about 1 to 2 cases per 100,000 per year. That translates roughly to about 5,000 new patients per year in the United States. And that number of 5,000 will be important in a little bit. The disease can affect any age group, but the average age of onset is somewhere between 50 to 60. And historically, people lived about 3 to 5 years. You got diagnosed, you went to the doctor said, you've got 3 to 5 years, and that was pretty much it. Okay, so now back to lessons learned. If we're going to do anything about this disease, we need to understand it. So breakthroughs require knowledge. You have to actually, hopefully you have to understand what you need to do. Breakthroughs require knowledge. So if we're going to intervene on chronic myeloid leukemia, we need to understand what drives the growth of this leukemia. And here we have 2 pathologists again. Peter Noel, David Hungerford, working in Philadelphia, in 1960 described an abnormal chromosome in the blood and bone marrow patients with CML. And they published this landmark article in the journal Science, which, for those of you not in the science field, this is the most prestigious journal in all of scientific literature. This is the entire article, 3 paragraphs long. That's right, quality and quantity do not equate. But when you publish a scientific article, in your last paragraph, you get to wax philosophically about the broad implications of your findings. And what they said were, the findings, this abnormal chromosome, suggest a causal relationship between the chromosome abnormality observed, and in those days it was called chronic granulocytic leukemia. Now, turns out they were absolutely right, but in 1960, nobody believed them. The general view was, this is some associated abnormality, has nothing to do with the cause, but they were absolutely right. So, next lesson. Breakthroughs often occur when different fields of investigation converge. And currently there's a lot of emphasis on what's called convergent science. But you also have to realize you have to apply the right technology to the right problem at the right time. And when I started out in the lab, if you could sequence an entire gene, that was huge. And it would take you weeks, if not months, to do one whole gene sequence. Today we talk about how many genomes can we sequence in how short a period of time. So if I wanted to talk about whole genome sequencing 20 years ago, that would have been crazy. So the reality is you have to do the right technology at the right time based on where you are, and that's part of the reason breakthroughs take time. In the case of CML, there were 3 threads that came together, 3 critically important threads. The field of tumor virology was started in around the early 1900s with Peyton Rouse at Rockefeller University, led to the oncogene field, broke open by Bishop and Varmus at UCSF, and that led to the molecular cause of chronic myeloid leukemia, this fusion gene called BCR-ABL. There's an entire field of chromosome analysis that identified this short chromosome in the patients with CML that lets you map genes to chromosome locations that allowed people to realize that 2 genes had broke apart, joined together, and created this abnormal fusion gene. And this entire field of protein phosphorylation, a post-translational modification of proteins that put phosphate onto specific residues, tyrosine kinase putting phosphate onto tyrosine residues, allowed you to begin to think about, could you develop a specific therapy for this molecular abnormality? So I got into this field right around 85, when all these things were coming together, and I looked at them. I was working on kinases at the time and thought, here's a disease I know is caused by a kinase. I should be working on it, and maybe I can move us this direction. So another thing I want to talk about is breakthroughs require seeing things differently. Now everyone says, oh, you've got to think outside the box. Well, I want to get you to think sometimes the answer is staring you right in the face and you can't see it. So I'm going to do an audience participation exercise, so get ready. I'm going to put a sentence up, and the rules are I want you to read the sentence from start to finish once through, and as you're reading the sentence, count the number of F's. Okay? Everybody ready? Okay, let's see how we did. How many people didn't, raise your hands, how many people didn't see any F's? How many people saw 1? How many people saw 2? How many people saw 3? How many people saw 4? How many people saw 5? How many people saw 6? How many people saw 7? How many people saw more than 7? Okay. Same rules. Read it through one more time, start to finish. Okay, how many people saw 1 F? 2, 3, 4, 5, 6, 7. Okay, go ahead and count. 6. We all read the same sentence, and the answer was right in front of us. Now, there are lots of reasons why this occurs this way, because lots of people drop the F's in of, but the reality is sometimes the answer is right in front of you, and you just have to see it for what it is. Now, I'm going to give you a couple of real-life examples of where that has happened, one non-medical and one medical. I'm going to start with Velcro. So Velcro, really useful invention. Turns out this guy, Georges de Mestral, a Swiss engineer, was hiking through the woods one day with his velvet vest on and his dog, and comes back and his velvet vest is covered with burrs. Now, how many millions of people have gone on hikes, come back with their clothes covered with burrs, picked the things off their clothes, had some choice words for it, and then went on with their business? Well, George said, why do these things stick so tightly to my velvet vest? He asked the critical question, why? Turns out he had a microscope down in his basement, so he went down and he saw that his vest had loops, the burrs had hooks. And he said, I bet I could make a fastener out of that. So again, something that anybody could have seen was right in front of him, and he made it into something useful. Alexander Fleming, a lot of people have heard the story. He walks into a lab, sees that his plate of bacteria are contaminated with yeast, and instead of throwing it out like millions of people have done, including myself, he looks at it a little bit more carefully and says, oh, isn't that interesting? The bacteria aren't growing very well around the yeast contamination. Maybe I should track that down. Well, the backstory of Alexander Fleming is that he is or was a surgeon. He had spent a fair bit of his time during World War I growing frustrated at taking bullets out of wounded soldiers only to have them die of infections several weeks later. So he dedicated his career to identifying specific ways of killing bacteria. And about 12 years after he had started in the laboratory was the fateful day that he walked into the laboratory and saw that contaminated plate of bacteria. So again, the answer was right in front of somebody, but they needed to see what was there. So I got into the field around 1988, 1990, and here is where the field was. For CML, we had this abnormal chromosome called the Philadelphia chromosome, because that's where Nolan Hungerford worked. It's present in virtually every single patient with CML. We knew from animal models, if you put this oncogene into mice, they get leukemia. And we know that it functions as an activated intracellular kinase. And one of the first laboratory experiments I did was I made a mutation in this kinase, made it kinase inactive, and it didn't work. So I saw this as pretty simple, straightforward. Shut this thing down, specific therapy. The rest of the world thought, oh, it's a much more complicated disease. It's not a simple disease caused by one gene, and targeting this probably won't work. So the way I looked at it was, this is how kinases worked. Over there on the left, kinases bind ATP. They transfer phosphate, in the case of tyrosine kinases, to tyrosine residues on proteins. And it's these tyrosine phosphorylated proteins that cause white cells to grow uncontrollably. So I thought, you shut that down, and you have a specific targeted therapy for CML. Turns out that there were a couple of companies. One was Novartis, it used to be Ciba-Geigy, and Nick Lydon was my colleague there. We worked together. His company, his group at Novartis had synthesized a handful of drugs that seemed to shut down a variety of kinases. And in 1993, they sent me a handful of their compounds for testing. And my lab showed that this one, which ultimately turned into imatinib or Gleevec, was the best of the drugs that they had sent me at specifically killing CML cells. Now, it should have been really simple. We had a drug, we had laboratory data, but there were lots and lots of reasons this drug never should have been developed. First of all, the prevailing view was you could never make a drug against a kinase. The prevailing view in the world was, I just showed you something that binds ATP. Well, in the genome, there are at least 500 to 1,000 proteins that bind ATP. And the view was ATP is ATP. You shut down one, you're going to shut down all of them. And why would you ever try to make a drug to hit one or two or three of these family of proteins? So the view was, it's just never going to be a viable target, so don't even bother. The next was, we're oncologists. And oncologists notoriously are a pretty pessimistic bunch. And the view was, when has a single drug in oncology ever worked? So why would you ever go after a single target in oncology? And then even biochemists looked at this and said, well, you're never going to swamp out ATP, so why bother with that as well? So we had all the oncologists against us and all the biochemists against us. And then we had the problem of toxicity. So even if we could get a drug against a couple of kinases, the view was the early knockout mice of individual kinases were embryonic lethal. So even if you shut down one kinase, the view was, you're going to kill people, and that's not a drug we ever want to take a risk on. But the biggest hurdle from the drug industry, 5,000 patients a year, how will you ever make enough money to justify its development? So if you think about this, the prevailing wisdom is it costs somewhere between $500 million to $1 billion to develop a drug, from preclinical on through marketing, and 1 in 10 drugs succeed. So if your market size is 5,000 a year, market projections were about 100 million a year, and a 1 in 10 chance of success. So if anyone wants to invest $1 billion for a return on investment of $100 million a year and a 1 in 10 chance of success, please let me know. We have lots of ideas for how you can invest those funds. But the view was this would just not make enough. Now, it turns out everything was wrong on this slide. And it said when you develop a successful drug, you just need one patient to tell you you've got a successful drug. And this was my one patient. And this is a before and after picture. Now, this is before imatinib and after. Now, the reason I have these numbers is the patient's wife was an accountant. She likes numbers, she likes charts and graphs. She'd come into clinic when her husband's white count was high, and she said, "Dr. Druker, you're not doing very well at controlling my husband's blood counts, are you?" And I'd say sheepishly, "Ma'am, I'm doing the very best I can," and I'd change his dose of medication around, and I'd usually overshoot. He'd bottom out, we'd back down, his counts would go up, and we had this seesawing pattern of his blood counts. In April of '99, we started him on imatinib at 300 milligrams per day, and by December of that year, she said, "Dr. Druker, you're actually doing pretty well. I'm going to stop checking up on you." Now, the FDA doesn't like anecdotes, and nor do I, but the reality is in 2.5 years from the first patient on our clinical trial to FDA, we got FDA approval. And it was one of the fastest FDA approvals in the history of the FDA, and still is the fastest from the time the drug company submitted the portfolio to the approval of this drug. And in 2001, it appeared on the cover of Time magazine with, is this the breakthrough we've been waiting for? So where are we now? So fast forward 8 years, we do a very large randomized trial, and I'm just showing you the imatinib or Gleevec arm. Now remember, 3 to 5 year life expectancy was historical here. Now with imatinib, 95% 5-year survival. And these curves are holding up out to 10 years and 11 years, still running about a 90% 10-year survival. So completely changed the course of this leukemia. And as Vic said, we have taken what was once a fatal or lethal leukemia and turned it into a manageable condition. But the reality is there are lots of people who are benefiting. And let me just share one story with you.
Hello, everyone. My name is Katie, and I'm very excited to be here tonight. I'm going to start out by telling you all a little bit about myself. I'm 18 years old. I'm a nursing student at University of Portland. I graduated in the top 8% of my high school class. Thank you. I was part of the Royal Crowns dance team where I was team captain, all-state athlete, and state champion. I love my dogs, Italian food, and Grey's Anatomy. And when I was 6 years old, I was diagnosed with chronic myeloid leukemia. We all know what happens in the treatment for cancer: chemo and radiation, hair loss and nausea. But would you like to know a secret? I never had any of that. I had Gleevec. Because of Brian Druker, my life is as I described it to you. Dr. Druker began developing Gleevec the same year that I was born, and it was FDA approved just 1 month before I was diagnosed. And if you want to talk about timing, that's some of the best that I've ever heard. It's because of the Knight Cancer Institute that this timing is possible. It's because of the Knight Cancer Institute that Dr. Druker was able to develop this groundbreaking new drug. It's because of Brian that I got to carry a first place trophy back to my dance team at state. And it's because of the Knight Cancer Institute that I'm going to school to become a nurse. Can you imagine a world where cancer is merely a memory? Brian Druker, Phil Knight, and you together are making this a reality, and I personally thank you.
So there are hundreds of other stories, thousands of other stories like this. And it's certainly extremely gratifying to see these things happening. But the reality is that this served very much as the groundwork for all of what we're hearing in personalized cancer therapy. It's about matching patients with the right therapy at the right time. And it started with Herceptin, it started with Gleevec, it started with many of these other drugs that showed oncologists that we actually can develop successful treatments based on the knowledge of what drives the growth of cancer. So where else has Gleevec worked? And it's worked in about a dozen different diseases, as Vic mentioned, the most prominent of which are gastrointestinal stromal tumor, a relatively uncommon intestinal tumor. About 2 or 3% of melanomas have a target for Gleevec, and a couple of other rare skin and blood diseases. So what lessons have we learned that we can now apply to other drugs? And I hope I've convinced you a little bit that you have to understand what you're doing. You need to have a good target. And if you have a good target and a good drug, you really can get good results. Now, I've used this slide for many, many years, and as you can tell, I put a lot of emphasis on the target. But recently, I've been reminded by a couple of near drug failures that you also need a good drug. There have been a couple of examples recently where there are some really good targets, and the example I'd use is BRAF in melanoma. There are a couple of drugs that came out that were called RAF inhibitors, but they weren't very good. They didn't work in metastatic melanoma, and then people said, oh, it must be the target. When other companies came in with really good mutant BRAF drugs, 60 to 80% response rate and some pretty good durability. So bad drugs almost kill good targets. So I have to balance this equation. You need both sides, good drugs, good targets, and you can get good results. So if we're going to translate the success of Gleevec to other cancers, we have to start by getting the right targets. These are going to be the early changes that cause the tumor. But we have to treat early in the course of the disease. If we use Gleevec for early stage disease, it works really well. If we get patients at much more advanced disease, it works, but people more likely to become resistant. So to do that, we have to develop reliable techniques for early detection. And these aren't going to be things that will detect things that don't need treatment. These are things that actually need treatment because we may deploy some toxic therapies. And we have to be smart about what we're doing, getting the right patient on the right drug. So if you have the right drug like Gleevec, we know what disease it will work for. We know what diseases it won't. We can be smarter about patient stratification. So where are we now? We're still in cancer in this kind of empirical world where we treat cancer because it's breast cancer, lung cancer, colon cancer. We use a lot of empiric treatments, and a lot of them are pretty toxic. And we never know who's going to respond and who won't. And we just can't predict what's going to happen. But that's changing. And with precision medicine, we're headed toward an era where we understand what's driving the growth of each and every cancer, and we treat based on that knowledge. And it doesn't matter if you have prostate cancer, breast cancer, lung cancer, we treat based on the targets. It's much like a mechanic lifting up the hood of a car and seeing there's an engine in whatever kind of car you drive, and the same parts will break. And we base the treatments on knowledge, the information, and the treatments can and will work the first time. They're going to be far more effective and far less toxic. And if you look at the spectrum of lung cancer, at least over half of the mutations in lung cancer are already known. And 10 years ago, my colleagues in lung cancer were the most pessimistic bunch, but now lung cancer is the poster child for precision medicine in all of oncology because of this knowledge driving the way that treatments are deployed. But in addition, as I said, deploying the right technology at the right time, what an amazing opportunity we have with the ability to interrogate tumors. We can do whole genome sequencing. We can do expression profiles. We can understand metabolomics, proteomics. And you guys here can put all this together and tell us in the clinic what to do about it. That's really exciting to me because this is all happening, and it's happening now, and it's allowing us to use precision treatments based on knowledge, not just empiricism. So I talked to you about where we were a century ago, and what do I see for this century? And I see us taking a very similar broad-based approach to cancer as we did for infectious diseases in the last century. And although I've focused a lot of my talk on specific therapies directed at critical targets, that's sort of like saying antibiotics are going to be all we need for infections. We know we need more. We know we need to understand and harness the power of the immune system, and I'm really pleased with what's happening here. Despite the fact that I've had this slide in my deck for over a decade, and it's been really hard to point to any success in the immune therapy, Now there are real successes. There are half a dozen different drugs targeting the ability of the immune system to kill cancer that are working, and they're working incredibly well. But in addition, we have to think about prevention and early diagnosis. Now, the reality is that for every $1,000 we spend on the first two, we're spending about $1 on these last prevention early detection. And in my view, even though my own institution is doing a lot of work in the specific therapies because of our leadership position, I worry a lot that we're not doing enough in the prevention early detection. So because of that, I started to put together a plan, and I started to seek investors. And if you know the landscape of Oregon, there's one real billionaire, Phil Knight, co-founder of Nike. And my institution, Cancer Institute, bears his name because of a $100 million donation he gave us about 6 or 7 years ago. So we began to put together a plan. We went back to Mr. Knight and his family and talked about what we might want to do. And to set this up, about 1.5 years ago, we held an event. He and his wife were the honorary co-chairs. And for those of you that know Phil Knight, he's incredibly shy. He doesn't like public speaking. The only way I got him to show up was say, "You don't have to say a word." A week in advance, he called me and said, "Is it okay if I introduce you?" Well, of course it's okay if you introduce me. Is there anything I can do to help you? Do you need my bio? And no, I'll just say a few things. So nobody in the audience had any clue what he was going to say, including me. But this is what he said.
As I speak, there is a grandfather, a mother, a child who hugs a loved one. Hugs a loved one he or she would not be able to hug if it were not for Brian Druker. It is incumbent on everyone in this room to do what he can to keep the miracles coming. And I speak to myself as well as to all of the rest of you. Accordingly, I make the following pledge. Penny and I will donate $500 million to OHSU if it is matched in pledges within 2 years in a fundraising campaign. If the campaign raises $499 million, we are relieved of our pledge. If it raises $500 million or $10 billion, our donation will be $500 million. Is there a higher calling than curing cancer?
So, 2 years, $500 million, all or nothing. To give you a little bit of context here, my cancer centers raised $5 or $10 million in a good year. So, seemingly impossible feat, but here's where we are with about 10 months to go. $482 million raised, including $200 from the state. This $200 from the state for buildings counts. Over time, we'll replace that, so it's about a $1.2 billion investment. Now, what was remarkable about the state was when he went to the state, it was, isn't this what you want your state to be known for? Aren't these the kinds of jobs that you want? And it was passed 85-5. Republicans and Democrats agreed on something. We've gone out to the business community. They've gotten behind it. We've gone to the labor community. They've gotten behind it. They had a campaign called Unite for the Night. We've had donors from all 50 states. We've had bake sales. We've had $50 donors. We've had $100 million donors. But people have rallied behind this, and they have put us close. But as Mr. Knight said, $499, we don't get anything from him, so we have to get to that finish line. So, you know, when I'm talking at Google, and $1 billion might seem like not a lot, but for academics, $1 billion is a ton. And so what we want to do, though, is we want to do something differently. We want to stop being a bunch of academics working on disparate projects. We want to make an impact. And so we want to think about how can we make an impact by putting together a team of people and focus them on a problem. So in a very short order, we want to assemble a team. We want to focus them. We don't want them spending all their time writing grants. We want them to focus on solving a problem. We want to equip them well, get them the right tools so they can actually focus on the work. We want them to work as a team. And I know, again, at a place like Google, teamwork seems so obvious, but in academics, we don't incentivize it. We incentivize people to do individual parts of a puzzle and hope it works out someday. Why don't we work as a team, focus on a problem, and solve the problem instead of making the problem, how are you going to get your next grant, or what are you going to publish? And we want to be open and sharing. If there are people around the world, around the country that can help us, we want to work with people that can help us. If some of the money needs to go outside of Oregon because somebody doesn't want to move there, we can do that. We want to solve a problem. And in addition, we're going to have to train a whole bunch of people because we don't think the workforce exists to do this work today, but we want this work to be sustained. So it's an ambitious plan. We're just getting started on that. We're planning our buildings. We're beginning to recruit people as we close in on our goal. But along the way, we've actually had some fun. So this is Gert Boyle, Columbia Sportswear. And she had this ad campaign several decades ago where she put her son under ice or throw snow on him with a bulldozer. And she was called One Tough Mother. Gert, actually, I worked with her sister as an undergraduate at UC San Diego, and Gert donated $100 million to our campaign. So we put together this thank you campaign with her. But more importantly, what Gert did was she inspired our most important people, and that's our patients. This is a patient who's going through a bone marrow transplant for her lymphoma, and she wanted to get on the action too, so she's got her One Tough Patient tattoo. So we have a lot of work to do. We have a lot of promise. We have a lot of potential. We need to live up to that. But I'm confident we can do that. And in closing, the people I really want to thank are the patients who went on this journey with me. These are people who had been told, get your affairs in order. You may have less than 6 months to live, and they found their way to our clinical trials, and they're still here today doing things they enjoy. This is a picture of the longest continuous patient on Gleevec. She is now 16.5 years from taking her last family trip because she thought she wasn't going to be around much longer. First patient from Italy doing things he loves, which is dancing. Patient spending time with their grandchildren. And this is actually a 14-year-old picture. She now has 2 great-grandchildren from the 2 older children on this picture. Lots of people who travel to Oregon for our clinical trials. And some people doing some absolutely remarkable things. This is a photo of a woman who came to Oregon in 2000, was the first patient from Australia treated with Gleevec. She went back to Sydney and was one of the torchbearers for the Sydney Olympic Games in 2000. So individual people, individual success, but it all starts to add up. And so we can take one disease at a time, one person at a time, and it all starts to add up. And this is my picture of hope, my hope for the future, that we see lots more pictures like this of people surviving and thriving despite a diagnosis of cancer. Thank you very much.
And Brian, maybe first I can ask, every generation, every 10 years or so, there is a war on cancer, a battle declared on cancer. Each generation has its own reasons to be optimistic about new scientific developments, things that happen in allied fields. And I know you mentioned a bit about this, but perhaps you could tell us why you are particularly optimistic this time that not a cure for cancer, but at least management of cancer as a chronic condition is within reach.
Yeah. So I don't know how much of the Emperor Maladies you'll show or you people have watched, but is so different than it was 20, 30 years ago when I started out. And we had no knowledge of what we were doing. It was all empiric. And when we think about hope, to me, this is real hope. And when you see rapid, dramatic responses because of an understanding of a cancer, as opposed to something that you have no clue what, how, or why it will work. And we really, to me, it's a matter of unraveling this puzzle. And so I think that actually it's a very different field. When I have lung cancer doctors who are optimistic about what they can do, or melanoma doctors, or the New York Times has an article about kidney cancer doctors having 5 drugs they can use when 5 years ago they had none. And oncologists are even becoming optimistic. And when that happens, there has to be some real hope. And I just think we're taking a completely different approach by understanding sort of these 3 pillars. In the past, it's always been, we've got to kill it, we've got to kill it, we've got to kill it. And now it's, yes, we have to kill it, but we have to kill it more intelligently. We have to bring in the immune system, and then we have to think about moving up more early. And so it's always knowledge driving it as opposed to this empiric, maybe we'll just combine the right drugs and get lucky.
Sure. And I do think in this audience, most people will be familiar with the new tools that are generating new information about the molecular basis of cancer and the new ontology of the disease that's resulting. Let's talk about the other 2 things that you mentioned. One, you called it the other side of the equation. Let's start with that first. I wonder if you could tell us about some new tools and techniques for manipulating cancer, manipulating the tumor environment, including immunotherapies.
So one of the things that we're understanding about cancer is that cancer isn't just this isolated tumor that grows on its own, and it's surrounded by lots of immune cells. It has a microenvironment. And the best analogy I ever heard was I was talking about this to a guy by the name of Wayne Drinkword, who owns a construction company. And he's a really bright guy. And he said, oh yeah, I go and I do accident assessments at my site. And sometimes it's a bad worker, and sometimes it's a bad environment that causes all my workplace accidents. So cancer isn't any different. Sometimes you'll get a bad seed driving the growth of the cancer, but sometimes it's the soil around the cancer or the inability of the immune system to attack and target it. And understanding cancer is a 3-dimensional system using either organized, growing in better reproducible mouse systems, allows us to do better interrogation of what's driving the growth of the cancer and what might work and what might not.
So, related question. I mean, we've known since before the time of Ames that there are some environmental factors which cause cancer. We also now know of many genetic factors, many genetic lesions which predispose one to cancer. Looking at an audience like this, how would you think about the genetic risk for cancer? What causes cancer, and how do you think about environmental risk? How has that changed with these new tools and techniques?
Well, I may give a longer answer, so bear with me.
Please.
First, I want to make a pitch for, we have to continue to recognize that we need to get people not to start smoking or to stop. It's still the #1 cause of cancer deaths in our country. But why is smoking so lethal? And it's lethal for 2 reasons. First of all, it directly damages DNA. And second of all, it injures lung tissue, so it causes the cells to grow. So it's a double hit. It causes your cell to grow, and when they grow, the DNA gets damaged. That causes mutations that are going to drive cancer. But that gives you a window into other environmental causes. Chronic inflammation that causes cells to grow more predisposes to cancer. Things that damage DNA, going to predispose to cancer. People born with BRCA defects that can't repair DNA mutations are more predisposed. So we can begin to get an insight into predispositions both genetically clearly all of us carry some predispositions genetically. You overlay that upon environmental factors, either through infections, exposure to infections, exposure to things that cause inflammation. We actually can take the same paradigm and move it back and say, if we understand what our predispositions are either genetically or at some point in time we see some reaction that causes some inflammation, we actually begin to get targets. So instead of this broad-based vitamin C, vitamin E, pick your favorite vitamin, or something is going to lower your chance of cancer. We're going to be able to actually go and target and say, if you take this medication for this period of time, we'll lower your risk of cancer. And we'll do it in a more precise way. I don't think we're quite ready for that, other than in very well-defined conditions. Sure. But we have the tools to understand that by doing sequencing, doing it again, and monitoring large cohorts of people to see what their outcomes are.
So you evoked inflammation again, and I want to return to the question about the immune response and modulating the immune response. But do you see the modulation of inflammation in a generic sense as a viable chemoprevention strategy?
I'm certainly open to considering general immune modulation as a potential for decreasing cancer risk. Part of it's going to mean how safe are the drugs, or would you look at high-risk populations, giving a window? If you see some inflammation, could you decrease that? Or in people, certainly with chronic inflammatory conditions, we know that, for example, chronic pancreatitis is one of the highest risk factors for pancreatic cancer. Now, there's a target population where if you wanted to decrease the risk of a deadly disease, that would be a population where I would think you could run a clinical trial. We know that ulcerative colitis, Crohn's disease, very high risk of colon cancer, and depending on where you are on the course of that disease, a colectomy may be a recommended course of treatment. I think we can do better than that. Right. And similarly with BRCA1, BRCA2, we recommend some pretty significant surgeries, mastectomies, oophorectomy. Could we think of a medical intervention? And again, if you start to extrapolate that all along that spectrum, we'll get to still high-risk conditions if we come up with the right genetic framework that say we might be able to target somebody, or if they have an inflammatory condition on top of that, maybe we intervene. And I'll give you one more example. I have an investigator who's looking at postpartum breast cancer, a really very highly lethal form of breast cancer, and it has to do with the change in the breasts that occur when women are pregnant and then either breastfeed or don't, and the changes that occur there. There may be a window or you could use an anti-inflammatory that would lower the risk of getting that lethal form of breast cancer. So we have already lots of clues there.
Interesting. And if there are any questions, please just stand up. Philip, the slide which showed 4 hurdles in the development of Gleevec, and he asks you to dive deep into one of them.
Yeah, so one of the more interesting stories was there was some animal toxicity with Gleevec in the early animal studies in rats and dogs. And I looked at the way the study was done and thought, well, wait a second. We'd give a therapy to people. We give toxic chemotherapy to people. And as an oncologist, I'm not afraid of a little bit of toxicity. And if I see some liver enzymes going up, we stop the drug. That doesn't concern me. But the people at the company were really worried about that. They didn't want to do anything. And because of the way the company was set up, they couldn't go and talk to the FDA to ask their advice. So I called up the FDA and I said, here's the toxicity package. What do you think? Oh, my goodness, that's more than we usually see. Why don't you send it through? So I called the company back, told them what I had done, and they were really upset with me. And nobody does this. This is not the way we do things here. But it was enough to get them to consider that maybe we could actually get this through the FDA to start a clinical trial. And the other story I'll tell is, and how did I finally convince the company? And for years I would approach the company when one of my patients would die, and I'd say, why don't you go into clinical trials? And what I realize now, that I didn't have kids at the time, and if I really to have kids, I still am struck by the fact when I ask my kids, what do you want for dinner? I don't know, what do we got? And then I say, well, would you like pizza or a hamburger? They'll make a decision. Or they may say, I don't want either one, but at least they'll make a decision, they'll make a choice. And when I say the drug kind of, what I learned was people aren't actually any different than kids, they're just more grown up. But they still make decisions the same way, and we give them an open-ended question, why don't you develop this drug? Oh, I don't know, we don't want to. Then when I gave them a choice, and I worked with Nick Lydon, who had actually now gone to a spin-out company, I said, would you develop this drug in a limited phase 1 trial, or would you license it out to Nick and me? Now they've got a choice. Now they can take it to a board and they can say, which one do you want to do? And they decided we want to run our own limited scale phase 1 because if it works, we want all the royalties and all the income off the drug. So they made a choice. So a really simple sort of aha moment that 20 years later I realized I did the right thing by giving them a choice.
It's interesting, just to follow up on that. I mean, you're of course famous for your perseverance over these bureaucratic obstacles. Has the environment in oncology drug development changed, or do we still face many of the same obstacles?
I sort of divide the world into those that get it and those that don't. There are some companies that are completely on board, and some companies are still stuck back 20 years ago and are looking at how much money we're going to make as opposed to, can we get this through quickly? Can we get to the market and then expand? And Gleevec, I don't get any royalties on Gleevec. It was already patented when it came into my lab against any tumor in a warm-blooded mammal. So there was nothing I was going to add to that patent. And Novartis' revenue was $4 billion last year on Gleevec. So they completely underestimated the market. And again, the point is if you can get something to clinic quickly and then expand, that's, to me, the way to go, and some companies really do get that.
So just to repeat the question, Joseph's question is about the idiosyncratic nature of Gleevec with respect to other targeted cancer therapies and that the response is durable.
That's why I say we've got to treat early in the course of the disease when diseases are less complex. I don't think we're going to win by targeting advanced cancer. We're going to win by treating it more early when it's less heterogeneous, when it's more treatable, more curable. And that's why I'm moving a significant effort that direction, because I'm tired of having to answer the question like this, which is, yes, we need to get, I think we might be okay with triple, quadruple combinations of the right targets, shutting down bypass mechanisms, but it's going to be really tough, and it's going to take decades to do that. At that same point, I want to move up earlier when the diseases look more like CML, because CML in its earliest stages is kind of a pre-malignancy that ultimately will convert to a fatal malignancy in 3 to 5 years. If I can move the clock back for other diseases, we can have more treatments like this.
What do you think are the most impressive new methods to study the pre-malignant to malignant transition in cancer?
I wish I had a great answer for that. I think it's going to require studying of the right patient populations. And by that I mean a large cohort of people followed over time to see what lesions, what abnormalities correlate with poor outcome. I think we can accelerate that in what I've talked about in Oregon, for example. Could we go and look at every man or woman who's died of breast or prostate cancer and ask the question, why? Did we not detect their cancer early enough? Okay, maybe that's a reason. But if we did detect it, if we didn't, what do we need? What could we have detected early that would have said, this is what we should be detecting in these patients who are still dying of their cancers? And can we learn from from samples that are already out there as to what we should be detecting? So I think there are certainly some early wins we might be able to get to. But what to detect, how to detect it, that's our next decade of work.
Well, what about new animal model systems or in vitro model systems? I know we were discussing some of this at lunch. What's your feeling about?
I may be a little controversial on this, but
Please.
My view of the best way to study human cancer is to study humans. And I don't think we have great animal models. I think animal models may be informative for certain questions regarding the microenvironment, because you can't do those studies in people. But if you want the genetic lesions, I think you've got to get people, because they're going to tell you what's happening. And we can artificially put them into animal models and say, oh, we have a model of pancreatic cancer. But then you've got to go and validate it in people anyway. So why not just skip that intermediate and go right to people?
So does your skepticism also extend to patient-derived xenografts, organoid models, some things that are hybrids between these approaches?
Patient-derived xenografts and organoids are things that we are moving to in our center, and I certainly believe in them because they allow us to recapitulate human cancer. But to come up with an artificial model that you say looks like smells like, but it still isn't human cancer. Again, I think our resources should be devoted to study of human cancer.
This raises another logical question about modeling the immune system in animals, which of course is fraught.
Right.
So let's go back to the question of immunotherapies. What do you think is the next step beyond just checkpoint inhibition or combinations of checkpoint inhibitors with other things? What's the next step in immunomodulation and control and re-engineering, if you will, of the immune system?
The biggest issue to me is we can't predict who will respond to our immunotherapeutics and who won't. And if we could understand that, we might be able to convert non-responders into responders, whether that be, is it mutational burden? Is it the inability of the T cells to infiltrate and then get activated? What is it about certain tumor types or certain patients? In melanoma, it's a 20 to 30% response rate, and some of them are incredibly durable. What about the other 60 to 70%? Why aren't they responding as well? So I think we need to understand who's who, and if we understand why, then we might be able to convert. And to me, that's the biggest hurdle in immune therapeutics right now.
Just the information and understanding. So other questions? Stephanie first. Stephanie asks you to contrast the different response rates or the durability of the response rates in two tumors, and she asks whether it's a function of the stage of diagnosis.
Yeah, so just for those of you that don't know just as well as, the response rate to imatinib with CML is quite durable responses, and as I showed, their survival curve is 85, 90%. With gastrointestinal stromal tumors, responses last on a median of 2 to 3 years, and then you'll see resistance. So the reasons for that, I'm not entirely clear. One possibility is that imatinib isn't quite as potent against the target in GIST as it is against the target in CML. The other is you typically are getting the tumors a little bit later in GIST than you do with CML, so that might kind of tie into that. But it also comes back to my comments about earlier. If you use adjuvant imatinib for 3 to 5 years, we actually are seeing survival benefits and significant survival benefits. So again, it may just be the biological heterogeneity by later detection, or as you catch these tumors earlier, we may have a bigger impact.
The question is that many cancers may have a genetic profile that is not targetable or even lacks a clear target or a known driver mutation, and how can we apply the same strategy to them?
So I think about most of the common tumors that have been sequenced in the Cancer Genome Atlas, most have a target. But something like ovarian cancer, it's almost all P53, which we can't target. So we may find targets, but we may not be able to target them yet. So I can't think, if you know, I can't think of an example.
I was thinking on your slide about the lung cancer, for example, the big chunk
yes,
is unknown.
Right, so the unknown chunk on my lung cancer slide, there's probably there's probably P53 in there, there's RB in there. Actually, what I put were known, targetable with current drugs. I didn't put, so the reality is we know a lot more than what we can do today. But that's why, although I want to close the loop, and thank you for allowing me to do that. 20 years ago, kinases weren't a druggable target. So now there are hundreds of kinase inhibitors in trials, dozens that have been FDA approved for an undruggable target. So P53, not druggable, well, that's today's technology. Maybe tomorrow somebody will figure out how to target it. There's a huge RAS initiative headed by Frank McCormick, formerly of UCSF, looking at RAS, which is one of the most commonly mutated targets across all cancer types. We currently don't have a good drug for it. So just because something is undruggable today doesn't mean we aren't trying or we won't have one someday.
And perhaps as we apply more pathway-based tools to analyzing these key signaling systems, there will be upstream regulators or other regulators that are targetable.
Exactly.
So related question, do you think that the TCGA data, which is, I think, the most comprehensive data set of its kind available now, do you think it's been mined to the extent that it can be for new targets?
Yeah, it's an interesting question. And a group that I've worked with actually mined TCGA data and identified some new chromosome translocations that were in that database, but just not recognized because of the algorithms. Right. I think the answer is yes. I suspect most of the low-hanging fruit's been mined, but there may be more with better, more sophisticated analyses. But I think what we're lacking are more samples. And I think as the reality is we're going to get hundreds of thousands of cancer genomes sequenced, expression profiling. I think we just need a richer data set along with better algorithms.
So one thing that we started to discuss is how much information you think is available just by understanding in ensemble the cells in a tumor, and how much is lost by not really having analytical techniques that resolve with spatial resolution the heterogeneity in all these measurements within the tumor. Perhaps you can comment on that.
Yeah, so one of the things that Joe Gray, who we recruited from Lawrence Berkeley, has a spatial systems biomedicine initiative where he really is trying to understand cancer as a three-dimensional system where, for example, Herceptin lives in these filaments on cancer cells that talk to other cancer cells. That may open up a whole new way of looking at breast cancer and how they communicate one to another or to their microenvironment. And whether that is going to turn into a therapeutic, I don't know, but it's just fascinating new biology as we understand cancer as a three-dimensional system as opposed to looking at it in a petri dish.
Yeah, I mean, it really seems, as Weinberg suggests, that the tumor has all the complexity of any other organ of the body.
Yes,
right?
Exactly right.
So Mark's question is about early detection and specifically the discordant funding available for early detection versus programs that target later stages in the cancer story.
Well, I blame two things on why there's so much funding for late-stage cancer. The first goes back to when I was in training and Hodgkin's disease was cured with 4 chemotherapy drugs, and the principles were non-overlapping toxicity, different mechanisms of action, combine the right 4 drugs and you cure a cancer. And everybody got on board and said, we're going to cure cancer by combining the right 4 drugs, and you got CHOP, and you got ABVD, and you got all these quadruple combinations, and it didn't happen. But we had all this funding going. And then I come along and I introduce a drug for a target, and everyone says, we're going to cure cancer with these targeted drugs, and then you get resistance. And now it's maybe you've got to do 3 or 4 drugs. So I take some of the blame for the fact that we're funding so much advanced cancer because people got optimistic. And we forgot that for decades, 15 years, I've been talking about targeted therapies, immune modulation, and prevention, early detection. You've got to have those 3 pillars if you're going to make an impact. But because of the way that our funding has gone, it's been, here's something right in front of us, this patient needs something, here's a drug that worked, and let's all get on board for that. So it's been an interesting evolution in my own thinking that we have to do things differently, but it's really hard to convince large funding agencies that you need to move.
I think of the whole subject of early detection in general. I don't know if you'd agree, but it seems like the scientific state of early detection is abysmal right now, except in a few cases. You have non-specific markers, they may or may not be actionable, or imaging modalities where they contrast with respect to the pathology you're trying to observe is minimal. So how are we going to change this practically?
Yeah, I absolutely agree that the state of the art right now in early detection is abysmal, and there are certainly some pockets, Stanford Canary Center just doing some amazing work. There are a few other places where some things are happening, but that's why we want to make this investment and put a stake in the ground of let's get a group together and figure out how to do this better. And if I had a blueprint for how we're going to do it, I'd tell you, but I don't. I just know that we've got to make an investment. We have other people around the world. Cancer Research UK is done a similar assessment and said, we've got to build this field. And we're going to build this together. We'll build it with Canary, with Cancer Research UK, and a few other like-minded partners, and train the next generation that can actually make an impact here.
So let me ask a proxy question then related to that. Is the Holy Grail for you a multifactorial blood test or something from a tumor biopsy sample that would help you stratify patients, stage disease, maybe in the blood test diagnose it earlier? or is it a non-invasive molecular imaging tool that could really be applied at a population scale? Which is it, or do you not know?
I think it's going to be a combination of lots of things. And so if you look at the most effective tool for lowering the death rate from cancer has been the Pap smear.
Yes.
So, you could imagine that maybe for some tumor types you need a scraping of, do you need a cheek swab for head and neck cancers? Colonoscopy is already quite effective but pretty invasive. Could you imagine a stool sample that would allow you to detect something that you might need to do a colonoscopy? Are there imaging technologies that would let you see a pancreatic cancer? Or is there a blood test you've got to do first to narrow the bin of people that would go through an imaging procedure so you're not spending all of our healthcare dollars on screening? So I don't think it's going to be a one-size-fits-all. It's going to be each different tumor type may require a different technology. As a holy grail, maybe there's something you'll find from the baseline study that says here's the transition from health to disease that says you might need to be more carefully screened. That would be a holy grail if you could have a really inexpensive screening technology that would let you say this is a person that should undergo more intensive screening. But I think that's a long ways off. It's going to need large numbers of people and large long-term follow-up.
And perhaps large tumor samples or samples of the pre-malignant state as well.
Exactly.
I think there were some other questions. Yes, I'm sorry, I don't know your name. Joanne's question is about technologies that seek to interrogate non-specifically the immune system through combinatorial arrays of peptides and their role in cancer diagnosis.
Yeah, and I guess we talked about this earlier. I'd even go beyond that to what are called theranostics, meaning could you attach something to an immune peptide that would recognize an early tumor and destroy it, or allow the immune system to destroy it. So then you can get away from this conundrum of, is it going to be lethal? Is it not? Do I need to take it out? Should I leave it in? Can I watch it? And maybe you could just let the immune system get activated, take it out at the same time it's detecting. That would be another holy grail of what we could identify.
But I think her question specifically is about using the immune system as a tool for surveillance and early diagnosis of disease.
Agreed.
And let me add to it. Do you think that can be done non-specifically in the sense that you have a chip that exposes random epitopes? And then also, can it be done specifically in the sense that can we learn enough about tumor antigens to actually design a test that uses the immune system to detect cancer?
I think you've stated perfectly what needs to be done, and I certainly believe that can be done.
Does Gleevec have a role in multiple myeloma, and do you conduct clinical trials for Gleevec or other agents in that disease?
So Gleevec was tried very briefly in myeloma, and unfortunately didn't have much benefit there. Myeloma is another field where things are changing very rapidly, and there are lots and lots of new agents being developed. There's an antibody against CD38, which is one of the cell surface markers on myeloma that's showing spectacular results in clinical trials. There are several new agents for resistant myeloma that are also showing remarkable activity and are getting moved up to earlier stages. We aren't a huge myeloma center, but there are several around the country that if they're asking for a specific person in your family, I'd be happy to help refer to some of these trials.
Let's speak briefly on a related subject about CART therapies, chimeric antigen receptor T-cell therapies, which has its antecedents in Star Trek. So it's something that we like in this audience. But how do you feel in general about the idea of re-engineering tools of the immune system to fight cancer?
Well, again, the CAR-Ts are, again, another example when I talk about immune modulation of the remarkable success of immune modulation. The concern I have with the CAR-Ts is that there's still a fair bit of toxicity to them, and we need to understand how to better utilize them in a safer manner. Some of my colleagues in bone marrow transplants say, well, we're used to doing bone marrow transplants and rescuing people, and we can do these cell therapies, and I agree, we can. It's just that having started out from a perspective of how can we treat cancer in a simpler, less toxic, less invasive fashion, I see the results from the CAR-Ts and I like what's happening, but I also think how can we do better and get them to a safer place where we can utilize them more effectively.
It's also one thing if the CAR T therapy is an ablative therapy against B cells,
Right.
where there's a well-defined target. But how optimistic are you that these things can be extended to solid tumors, where I guess it faces the same problem that we're discussing in the molecular imaging context?
Again, those are my concerns. If you're taking out an entire cell type, B cells you can kind of live without, and you take out some other cells that you need for heart, lung, these could be really toxic. And even as we move to myeloid malignancy, some of my colleagues have been unwilling to take out the myeloid lineage because you're going to be predisposed to infections. So we need to understand how best to target, what the good targets are, and maybe how to even dial back on some of the toxicities.
His question, to paraphrase, is that some argue that Gleevec is curing the disease it treats, and then at what point do you stop treatment, and how do you, I think, ethically think about doing that?
So about 20%, maybe 30% of people on Gleevec will be molecularly undetectable for the BCR-ABL transcripts. And if you stop therapy, 50% will remain undetectable and 50% will relapse and need to be retreated. So it's 50-50 of the 20 to 30%. So it's a small percent, and we don't know who's who. So there's some hints that it's immunologic. And the hints are as follows. We know that people who previously received interferon, which is the prior generation of treatment for CML, have a little bit higher chance of getting off therapy. And I've had patients who I've taken off therapy, their PCRs for the BCR-ABL transcripts turn slightly positive. I'll check again and they're back to undetectable. And they bounce around at really low level, and they've done that for years. So I can't imagine, we haven't eliminated the last CML cell. So to me, it means there's got to be some immune recognition. Now, if we can take those clues and say, what is it about the immune system that's being recognized on the CML cells, and can we convert people from unable to discontinue to being able to discontinue, then maybe we can actually get more people to stop and get from control to cure. And that's actually one of the current areas of focus in my lab.
So in several answers, several responses, including this one, we kind of take for granted as a thought experiment that we'd be able to recruit the cohorts to do these gigantic trials of combination therapies. But the lamentable fact is that only 3% of patients are in trials to begin with in oncology. So how optimistic are you that these questions can actually be answered today?
Yeah. So it's lamentable that we enroll so few people in our clinical trials. And in part, that's an infrastructure issue, meaning we just don't have enough people in the healthcare industry to put people on trials. I don't think it's a lack of interest. I think it's just a lack of enough manpower to do the work. So I think that's going to require some investments. And I didn't go through all of our billion-dollar plan, but I'm putting about $100 million into our clinical trials infrastructure so we can put more people on trials at our institution. But it's not going to get us to the tens of thousands of people we need to actually test with these combinations and doing them quickly. But if we can show that it can be done at one place, maybe other places will follow.
And perhaps there is some overlap in the technologies one would develop for early detection and the ones you would use for stratification to design better powered basket trials and other trials, biomarker driven.
And certainly in AML, that's the other area we're putting a lot of investment is we're doing a basket trial where everybody gets screened. We have treatments for everybody. So our idea is anyone with leukemia gets enrolled on a clinical trial. And it should be 80% of people, not 3%.
As it is in the childhood analog of these cases. So the question asks about a global perspective on cancer and how can some of the, I assume, advanced technologies that we've been discussing actually bear on the global problem.
Yeah. So I'm going to come back to an earlier statement. First of all, one of the things the U.S. has done really well is export smoking. And so a lot of the cancer deaths worldwide are smoking related, and it's not something we should be terribly proud of, but that's, we talk about prevention, we have to think about how we manage that part. As far as how we manage advanced cancers, and if you look at the paradigm in HIV, it is a really tough road to think about managing HIV in countries that they can't afford the drugs. And so that's why I think we've got to think about, we have to imagine how do we do early detection that's cost effective? How would you pap smears in around the world? And there are certainly technologies that can allow us to do that, have them read at a central location, and can lower the death rate from cervical cancer. So there are already programs in place that can be the leading edge, if you will. But I think the best way is going to be prevention and early detection. If we're going to rely on this safety net of treating advanced cancer, we're going to bankrupt entire countries by doing that. So globally, we've really, to my way of thinking, this is where we need to be moving. And it's the same as you think about HIV. It's vaccination that's is really going to be the best technology at making a global impact on HIV. Same with malaria and other diseases. We've got to think about how you do this cost effectively.
So speaking of vaccination, I think we'd be remiss to not ask you to comment on HPV vaccination and the controversy it's caused, at least in a few U.S. states. What's your feeling?
Well, my kids have all been vaccinated, boys and girls. So I hope that speaks for what I think about it. I'm acting in the way that I think a responsible parent should. And it's not about, you can make it about whatever you want, but for me it's about preventing cancer. And early on I had thought about, do we put legislation through in our state? And after seeing the pushback from other states where it just didn't happen, I just realized we're not going to win this battle. But I can only speak for my own family and what my beliefs are.
Yeah, absolutely, and I think in that case it is such a clear example of chemo prevention, if you will, that it is. Yeah,
and before some of the immune therapeutics came along, that was what I put in the checkbox of immune modulation, was a vaccine that can prevent cancer. Now, don't we want more of them?
As we develop new treatments, and I think it's true broadly of any treatment, including in HIV and infectious diseases, there will be some societies that find them objectionable for cost or other reasons. In the case of HPV, how do we deal with that?
Yeah, we've got to engage our social science colleagues on this, and we have to figure out these problems. Because you look at any societal problems, it's often as much behavioral modification as it is anything we can do medically, and I don't have a clear view on how we accomplish that, other than we're going to have to face those issues.
So in the end, the solution might be web advertising.
It very well could be, or it could be social pressures. And you look at how do you get people to vote? Did you post on your Facebook page and I voted, and I want all my friends to vote. I stopped smoking, I want all my friends to stop. I got the HPV vaccine, I want all my friends to. There's incredible pressure with social dynamics, and we need to harness that.
So one last question, I think then we have to start the documentary, but I know you were visiting our colleagues at Facebook this morning. So tell us what the responsibility and the duty of the Silicon Valley technology industry is in this area, in early detection. And what, in your opinion, are the unique tools, if any, that we've developed that could help you with this problem?
So I'm going to take two points there. First of all, I think everyone saw the power of philanthropy, and the reality is that there is a lot that can be done with philanthropy in moving things forward that need to be moved forward. So I think it's important to recognize, the people that run Google, the people that run Facebook are giving a lot. And that needs to be encouraged, because there are societal problems. It's not just throwing money at problems. It's actually throwing money at smart problems, getting to solutions. But the sorts of tools that are being created at Google, particularly in the ability to understand biology by bringing large amounts of data together, aggregating large amounts of data, and then helping us understand what that data is trying to tell us. When I started out in my career, my first mentor said, you've got to let the data tell you what it's trying to tell you. Let the data speak for itself. And I think that's what Google is doing so well, is bringing all this data together and letting it speak. And so you can identify patterns, and you do so well at identifying patterns and what is the most important, because we're going to be deluged with data. But we need to understand that. So it's not just as much generating it, it's helping us figure out, sifting through mounds of data, bringing it together, identifying the patterns, and then saying, that's what we need to go. And that's where the tech industry can help us. It's bringing computational biology, it's bringing pattern recognition, it's bringing computational algorithms. I don't think generation of data is our limiting factor right now. I think it's understanding what it's trying to tell us.
And please join me in thanking Brian.
Thank you.